In mathematics in particular, we think it's wise to credit our co-authors at the beginning of the lecture before everybody falls asleep. And also for reasons I can't quite put my finger on, we don't usually show photographs of our co-authors. So this work's joined with Nick. And it's part of a rather amorphous collaboration that we've had going for a long time. And what I'm going to talk about involves stuff done by Jerome Kelleher, who's a computer scientist, which is the only computing aspect of this talk, Daniel Straulino, Amandine Weber, and Feng Yu. And all of these people have either been graduate students or postdocs in Oxford in the last few years. So what I want to talk about is a bit different from what you've been hearing about. I want to talk about the interaction between selection and genetic drift for populations that are living in a spatial continuum. And in order to do that, of course, the first thing I need is a decent model for genetic drift in a spatial continuum, which is not something that's so easy to get a handle on. So the first half of the talk is actually going to be telling you about that. And apologies to people who've seen me talk about this before. So the basic motivation, so this was the creature that Nick was very excited by when we first met in the mid-1990s, Pedisma pedestris. And what was pointed out to me was that most of the classical models of population genetics incorporate spatial structure by supposing that the population is subdivided into demes living at the vertices of a graph. And one's individuals in a population can move around by moving only along the edges of that graph. Now, this is where Pedisma pedestris lives. I stole this from the internet. I think it's from Richard Nichols' website. And try as I might, I cannot see this as a nice discrete graph. So the question is, can we actually model directly populations in a spatial continuum? So the mathematical challenge that was posed to me a long time ago by Nick was to find a consistent forward in time model for the way that the population evolves, but of course a backwards in time model for the genealogical trees relating individuals in a sample from the population, so that we could in principle compare to data. I can assure you there will be no data in this talk, but we could in principle compare to data. So Malako and Wright almost solved this problem in the 1940s, right? So they supposed that their population, they were working with populations distributed on the whole of two-dimensional Euclidean space, or one-dimensional Euclidean space, but we'll stick with 2 dimensions. And they supposed the initial population was distributed as a Poisson point process with constant intensity. What does that mean? It just means if you look in any region, the number of individuals living there is a Poisson random variable with a parameter proportional to the area of the region, and you can think of those individuals as just having been thrown down uniformly on that region. Okay? So you've got something that's very evenly distributed somehow over the plane. And their population evolved in discrete generations, and each individual leaves behind a Poisson number of offspring. They like Poisson, with mean 1. And the offspring choose their location just according to an independent Gaussian pick around the location of the parent. And not unreasonably, when you look at it, Malako and Wright assumed that this Poisson distribution that I started with, this nice constant intensity distribution I started with, would be preserved by this dynamic. So my population would always look nice and evenly spread across space. And under that assumption, of course, they didn't have access to DNA in those days, but they did talk about the probability of identity. So under that assumption, they were able to write down a recursion for the probability of identity of 2 individuals sampled at a given distance from this population. And this leads to, this is assuming that individuals in each individual in each generation has some small probability of mutating to a type different from its parent. Okay? And this leads to the Wright-Malako formula, which is a very powerful thing. But unfortunately, Felsenstein observed that there's a bit of an issue with the Wright-Malako model, or rather the Wright-Malako assumptions. An easy way to see it is to actually look at a simulation. So this is something Jerome did. Any nice pictures are due to Jerome. Remember the word nice. So he started with a large torus, and he's thrown down 1,000 points. So my Poisson process happened to have 1,000 individuals in it, and he's just thrown them down uniformly at random, right? So there's my Poisson starting condition. And then he allows the Wright-Malako dynamics to go on. So each individual in each generation leaves behind a Poisson 1 number of offspring, and they just choose a location according to a Gaussian distribution around the location of the parent. And after 10 generations, this is what it looks like. So the population size is still pretty close to 1,000. It's not really changed very much. So that bit's all right. But it doesn't look so evenly spread anymore, right? There's a lot of white space beginning to appear. After 100 generations, population size still pretty close to 1,000, but lots of white space. And if you remember we were working on a torus, you can see we've really only got one large clump appearing here. And after 1,000 generations, my population has noticed that actually the total size is given by a critical branching process, so it better die out sooner or later. And it's got down to 300, and not long after this it really does die out. But you can see that at this point it really is just one cluster, right? It's become very, very clustered. So Felsenstein observed this clumping effect on all of R1 or R2, and he said, well, okay, maybe we should work on a torus. Maybe it's because we're working on all of Euclidean space, and nothing lives on all of Euclidean space. But we can see that that doesn't work because we've still got this difficulty. And also, the population size is going to be a critical branching process, so eventually it's going to die out. And so that's all right. Let's specify. Let's suppose that somehow the total population size is exogenously specified. Well, we can see that's not going to work either, because if we look back at what happened at times 10 and 100, the population size really hadn't changed very much. If I conditioned my population size to be 1,000, I'd still get the same picture. I'm still getting this clumping phenomenon. And at that point, he gave up, and he famously wrote a very famous paper in 1975 and dubbed the problem the pain in the torus. And the lesson that we learn from the pain in the torus is that if we assume individuals in our population somehow reproduce independently of one another and they don't sense the local population density, then inevitably we get clumping. So if we want to overcome that clumping, our models have to have some sort of local regulation. And the classical stepping stone model has exactly that, because it assumes the population size in each deme is fixed. And that's the strongest local regulation you could think of. But we'd like to do something that's actually in a spatial continuum, which also has a local regulation. Now, there's a huge mathematical literature on this, and lots of people have proposed models of varying degrees of complexity that incorporate local regulation and have some sort of stationary distribution. But the problem is that analytically they're completely intractable. And in particular, we're unable to write down what those genealogical trees that we want to compare our models to are actually going to look like. Okay, so. Mm-hmm.
Why do we want to get rid of the clumping? I mean, is that not consistent with what actually happens?
Those clumps become arbitrarily dense and arbitrarily big. So actually your population really piles up. And so you want something that you really want some kind of a stationary distribution over the timescales you're looking at.
Okay. So there has to be some kind of individual repulsion or something.
There has to be either repulsion, or the most natural thing to do is to suppose that the reproductive success of somebody living in a crowded region is lower than the reproductive success of somebody who's living somewhere with a lot of resources.
Right, density dependent.
Yeah, okay. Density dependent regulation, absolutely. Okay, so here's a really, really naive model to illustrate how we can actually arrive at something which is analytically tractable. And may I reassure you that actually lots of other local structures would lead to the same limit that I'm going to describe. But let's pretend we're right in Malloco, and let's try and do everything based on Poisson processes. So I'm going to start with a Poisson process of constant intensity, exactly like right in Malloco. But now, reproduction is not going to be based on individuals, it's going to be based on events. And this is the key way to overcome the pain in the torus. So I have to have some formulae because I'm a mathematician, but so at some rate, I throw points down. I'm going to throw balls down onto the plane, or throw disks down onto the plane. And that rate is driven by what we call a Poisson point process. When an event, I'm going to do it in such a way that everything's translation invariant and time homogeneous. When an event is thrown down, when one of these disks is thrown down, if there's no one living there, I don't do anything. But if there are individuals living there, this is what I do. So if I had a more modern computer, I'd also have a pointer. First of all, I choose a parent uniformly at random from the individuals that were in the ball. So I've colored that parent magenta. Okay. And then each individual notice, first of all, that your chance of being picked as parent is inversely proportional to the local population density, right? So your chance of being a parent in the event is reduced if you're living in a crowded region. Now, I'm going to pick independently every time an event falls a probability. And that probability, U, I've denoted it, is going to tell me the proportion of individuals that are going to be replaced in this reproduction event. So each individual is going to flip a coin that comes up heads with probability U, and if it gets a head, it dies. Okay? So it thins the population. And then I'm going to throw down new individuals of the same type as the parent. These are they. And they're going to be born in order to replenish this Poisson process. They're also going to be born according to a Poisson process, just within the ball, no one outside the ball. And because I will have removed approximately lambda u times the area of the ball individuals, I will replace by lambda u times the area of the ball individuals. Okay? And I think there's one more. There we are. I've killed the other ones. And this is the state after the reproduction event. Now, if I allowed birth from nowhere, so if I hadn't said, if a region is empty, do nothing, but instead I've said, if a region is empty, I'll still allow myself to throw down some offspring, then this process would have the distribution I started with, the Poisson point process with constant intensity, as a stationary distribution. So this is as close to Wright-Mallico as you could possibly want to be. However, because I insist that you can't have births from nowhere, the Poisson process isn't a stationary distribution, but it almost is, right? So if lambda's big enough, this thing does have a nice sort of stationary distribution. We don't get uncontrolled clumping, and we can write down the genealogical trees. But the problem is, again, because regions can and do become empty, those are slightly cumbersome expressions that we get for the genealogical trees. But when you examine them closely, you realize that actually, if you forgot about the possibility of things getting empty, or if mathematically you let this lambda go to infinity so that regions never do become empty, they're very, very stable. So the genealogical trees for a moderate value of lambda, so for example, so that the average number of individuals in a ball would be about 5, are extraordinarily close to the genealogical trees that I get if I actually had an infinite intensity. And because it's really the genealogical trees I'm interested in, we're going to pass to the lambda equals infinity limit. Right, okay, so I'm just trying to compete with Nick for complicated expressions. So now, the object we're going to look at is what mathematically you obtain by letting lambda tend to infinity. And what it does is at each time t and point x, it specifies a probability measure on a type space. So K here is just a space of types, and in a moment that's just going to be 2 types. And the interpretation is, if I were able to sample an individual from the point X at time t, their type would be determined by taking a pick from that probability measure rho of tx. So it's just telling us the distribution of types in our population as time goes forwards. And how does it change with time? Well, again, I use this Poisson point process of events. So I throw down disks in a homogeneous way at a constant rate. And when a disk falls, first of all, I'm going to pick a location for the parent. That's just going to be uniformly distributed on the disk, because I don't know where individuals are. Lambda's infinite now. So they're sort of evenly spread everywhere across here. So the location of the parent is going to be uniform. I ascertain the type of the parent by taking a pick from the probability measure at the point where the parent was. This was the point z, immediately before the event, right? That gives me a type. And that tells me that all offspring of this event were of type k. So I have to update my sampling probabilities. So for each point in this ball, each point y in this ball, immediately after the event, if I sample an individual, the probability they were an offspring is u. And therefore their type is k. And that's what this means. The probability they were not an offspring, that they were already there and they just survived the event, is 1 minus u. And then their distribution is determined by sampling as if the event had never happened. And this gives us a way of modeling the way allele frequencies evolve forwards in time. Okay. Well, that's kind of complex stuff, but it turns out that the backwards in time process of genealogies is actually a little bit easier to get your head around. So again, I'm showing off. This is proving I'm a mathematician and can use LaTeX. Let's think about a single ancestral lineage. So if I take an individual, I sample an individual from the population, nothing's going to happen to it until it's covered by one of these reproduction events. When that happens, it has probability u of being an offspring of the event. And if it's an offspring of the event, its parent was at the location of the parent of the event, which is just a uniformly distributed point on the event. And that means it's actually a single lineage. It's just going to move around according to a continuous time and space random walk. It's just going to jump to whenever it's hit by a reproduction event, it jumps to the location of the parent. And that's something I can write down exactly the distribution of, and that's all this says. Okay? But different lineages are not going to evolve independently of one another. And in particular, lineages can coalesce when they're hit by the same event. So this is meant to, all the green circles are meant to represent lineages immediately before the event, as I trace backwards in time. Lineages not hit by the event don't change, but if this event encompasses a number of ancestral lineages, I can have multiple lineages being offspring of the event and therefore merging into the common parent. So I get coalescence events when individuals are encompassed by the same reproduction event, and those coalescence events may not be Kingman-like, it may not just be pairwise, it may be multiple mergers. Okay? So what you've got is a system of random walkers jumping around according to continuous time, random walks with continuous jump size, with continuous distribution of jump size as well. And when they're encompassed by the same event, so when they're close enough together to be hit by the same event, they have some chance of coalescing. And that turns out to be a very analytically tractable process that we can study, and we can use it to say a great deal about allele frequencies in the population. Now, I should emphasize that, you know, I've presented this with disks and in a very sort of naive sort of way, always replacing things uniformly across the disk. It's a framework. What's important is that reproduction is based on events, not on individuals. As soon as you've done that, you've overcome the pain in the torus, and then you can do whatever you like. But the sort of results I'm interested in are going to be robust to whatever your choice of local reproduction mechanism is. I'm not pretending this is how populations evolve, but I am pretending it picks up some of the key features of the way populations evolve. Okay, and I told you there'll be one bit of data in the form of a picture at the end if I have time. Okay, so I wanted to investigate, remember my interest was in investigating the interaction between genetic drift, and now this is meant to be a model of genetic drift for a population in a spatial continuum, so I got our neutral model, and I wanted to look at the interaction of that with selection. So I'm going to take a very simple form of selection. I'm going to suppose there are lots and lots of ways I could do it. I'm going to suppose my population has just two types, little a and capital A. And then instead of thinking in terms of probability measures at each point in space, I can just specify what's the proportion of my, if I sample an individual from x at time t, the probability it's type little a is going to be written w of tx, but we won't use the notation very much. And to introduce selection, I'm just going to weight the way in which I choose the parent. Right? So the standard sort of way you introduce selection into a Moran model that I guess we've heard about already this afternoon, even if we didn't go to boot camp. So I'm going to insist, when I have a reproduction event, up till now, I've just sampled an individual uniformly at random from all those present in the ball. But now I'm going to give the capital A guys a weight 1 plus S, and the little a guys a weight 1. And S here is just a small positive parameter, right? Just a standard way we introduce selection in the Moran model. So if a reproduction event affects a ball centered on X and of radius R, and at the time when the reproduction event hits, the proportion of little a alleles in that ball is W bar, then the probability of the offspring, or of type little a, is going to be just given by this ratio. Okay. Now, S is going to be small, but you're only meant to see one at a time. S is meant to be small, so I might as well expand this guy as a geometric series, and I'm going to throw away terms of order S squared. And then this is kind of nice. It means when an event falls, I can think of it as being of one of two types. So I can say, well, with probability 1 minus S, my event is like my neutral events that I had before, and the chance of choosing a little a offspring is just W bar. On the other hand, with probability S, it's a selective event, and then the chance of having a little a offspring is W bar squared. And how am I going to make that happen? Well, I'm going to sample two potential parents, and only if they're both type little a do I have little a offspring. And then I just choose one of those potential parents at random. If one of them or two of them are type capital A, then I choose at random among the capital A's for the parent. Okay? So that just makes sure that I have this probability of sampling, and it also gives me a choice of parent. A nice thing about working with the Poisson process is that actually then we can think of selection, the neutral events and selective events, as being driven by two independent Poisson processes, neutral events falling at rate 1 minus S, and selective events falling at a rate proportional to S. Okay? So let's see Right, I'm going to be very brave and see if my computer's feeling up to playing a video. So, a quick pair if you're that way inclined. So here's the little a population. We seeded it with some capital A guys, and this is just how a selectively advantageous allele will spread out. And you can see that it gradually develops into what you'd expect, an expanding wave, right? From the point of view of the genealogies, all the interesting stuff is happening around here, right? That's where we're going to get interesting effects from the selection. And now, unfortunately, you can see we're on a torus and we've spilled over the edge. Okay. But that's, how do you stop this thing? Once it's started, it won't stop, right? But that's the sort of picture we get for the way that the allele frequencies spread out. Now, I said all the interesting genealogical stuff is going to arise because of what happens when we're in this noisy, very noisy wavefront here. Things get a little bit confounded by the geometry when you look at a mutation that's then sort of spreading in a circular wave. So to make our lives easier, we're going to think about a flat wave. We're actually not going to solve the problem of the genealogy, but I just want to show you the kind of thing that happens. So remember I said all the nice pictures were due to Jerome. This beauty is due to Nick. So we're not quite sure what it's of. I did ask him yesterday. But the idea is that this is a wavefront that progressed in this direction. I assume that these are coloured at the end.
Artistic reasons.
A bit for artistic reasons. And this is meant to give you some idea of where individuals were descended from. So actually, this was a stepping stone model. It wasn't the model I just showed you. And what you can see is that you get sort of a lot of lineages, a lot of the stuff in the front tugged into small numbers of kind of bottlenecks as you go backwards in time. And actually, I think we're actually perilously close to understanding exactly how to model this. But what you can see is that things are non-trivial. Things are really quite complicated. All right, so I can't tell you exactly how to solve that. But how would we? I mean, how do you try and get a hold of the genealogy during a selective sweep? Well, if you're Coop and Griffiths or Kaplan and Hudson, or indeed us some years ago, you try and look at the way that the allele frequencies evolve as you go backwards in time, and you do a coalescent in a random background. Can't do that here because we can't reverse the process of allele frequencies. In a spatial context, that's very much harder. We don't have a good enough understanding, in fact, of the way that the wave is going forwards in time to be able to say very much about what it does backwards in time. The other sort of the methodology that Bob Griffiths and I pushed, and also Donnelly and Stevens, I think, was using a kind of proxy for allele frequencies as you go backwards in time, which we call virtual lineages. Again, we can't do it because we don't know enough about the distribution of allele frequencies as you go forwards and backwards in time and how they evolve. So I was reminded by Conrad, who's probably here somewhere, that Nick and I have an ancient manuscript called Distinguishing Causes of Reduced Genetic Diversity, which we never published. And I think the reason we never published it was because we realized we simply couldn't. And so what the question that I decided that I would try and solve instead with a graduate student, Daniel Stralino, was to try and understand when there would be any patterns that we could actually see in our data. And to do that, we're going to use something which you'll probably throw up your hands in horror over, because it's a spatial analog of the ancestral selection graph. Now, the ancestral selection graph isn't terribly practical, even when there's no space, because the number of lineages can grow very, very quickly in the ancestral selection graph. So you can imagine it's even worse in space. But one of the advantages of hiring someone like Jerome Kelleher is he can actually simulate these things. But we're just going to use it as an analytic tool. So what's it going to look like? So what the ancestral selection graph is going to do for us is it's going to at least include the ancestry of a sample from my population. So remember what the genealogical trees looked like in the neutral case. We had individuals move around according to random walks, and when they were encompassed by the same event, then they could coalesce, right? Well, the neutral events that we have, and we've split our events now into two sorts, neutral events and selective events. So neutral events, that's fine, it's exactly as before. But at a selective event, suppose I'm just looking at a single lineage at a selective event, I can't decide which of the two potential parents I picked is the real parent unless I know their types. And to find out their types, I'm going to have to trace further back in time until I've got some idea what the distribution of types in the population is. So what I do, it's entirely analogous to the ancestral selection graph that Krohn and Neuhauser introduced in the absence of space, is that when I get to a selective event, I follow two potential ancestral lineages. And then I go back to some time in the past, and I can resolve and assign types to the ancestral potential ancestral lineages that are around, and then I can move back through the tree, resolving who was actually the parent at each of these events. So here we have an event where these guys were all offspring of the event, but I replaced them not by one lineage as we did for the neutral events, but by two. Okay? So I have a branching and coalescing structure. And one thing that makes this kind of fun, actually, is that at least if I want to know things like what's the probability a sampled individual is type little a, well, only if and only if all the lineages in this set of potential ancestors are of type little a at every previous time. And that actually allows me to say things about the way that the allele frequency spread forwards in space. But let's use it to answer this question, when have I got any hope of actually seeing a selective sweep? So, of course, if the selective sweep sweeps through a population, we no longer have any variability at the selected locus, and so we're going to try and detect it by looking for patterns of neutral variation close by. And that means that if I'm going to see the effect of selection, those patterns of neutral variation have to be distorted, right? And neutral variation is then going to set the time scale for the sort of coalescent process that I'm looking at. So I need that the coalescent process, let's suppose there's no recombination, forget recombination. So suppose that the coalescent at the selected locus is going to, suppose I'm going to see any interesting patterns at the neutral locus, I need my coalescent at the selected locus to look different from what it would have done under neutrality, in the sense that when I look at it, when I look for neutral mutations on it, I see something different. So that means when I look at it over timescales that are set by the neutral mutation rate, so timescales of order 1 over mu, it's got to look different. Now, in a spatial context, those timescales also dictate a spatial scale, right? So if I'm looking at ancestry over a timescale of order 1 over mu, and asking, is it looking any different from the neutral case, then the spatial scales over which I expect to be looking, well, remember a single ancestral lineage was basically following a random walk, so I have to look over spatial scales of the order 1 over square root of mu. So that's what we're going to do. We're going to actually look at our ancestral selection graph over timescales 1 over mu and spatial scales 1 over root mu. And just this comment that because mutation rates are going to be low, this means I'm looking at large temporal and spatial scales. And scaling limits, so what we're actually going to do is look at a scaling limit, but scaling limits are wonderfully robust. So the fact I took this very special model with disk replacement and all this kind of stuff isn't going to matter. There is a vast array of other models which incorporate the same basic getting over the pain in the torus and a bit of selection, which would give you the same limit. Okay?
So I guess I'm confused why the scale is 1 over square root of mu, since you have this propagating wave associated with selection.
Well, because I'm really looking at a neutral locus, right? So that you're interested, I mean, where are you going to see most of it? Anyway, look at your Gaussian. I mean, you're going to see everything on square root of mu.
But the wave sweeps out, right?
Yeah. But that's not all the. But the you'll see that you won't see. I mean, if you don't see anything on this scale, you won't see it on a longer scale. The density of the wave at the front. So okay, your rightmost particle is spreading out a long way, but most of your wave is still focused around wherever it began. So you're still going to see the interesting patterns. You're going to look at where you would expect to see your neutral mutation, and you're going to see most of the patterns are going to be, think about a single selectively advantageous mutation arising. You've got this Gaussian. Yes, you're going to see somebody way out here, but most guys are still going to be in here. So we can expect high density. If you actually do the calculation with your logistic control and so on, most of it is still looking like a Brownian motion around where you began. It's just it's going to be at higher frequency there. So we can talk about it. Okay. So we're going to look at this limit under small population density. So what do I mean by that? The classical, we'll do high population density in a minute. Small population density is just saying if I'm looking in a spatial continuum, then the number of individuals living in one of these regions is not necessarily going to be particularly big. In particular, it's not going to be infinite, which is what the standard deterministic differential equation models assume. So what I'm going to do is I'm going to fix the proportion of individuals that are replaced in an event at something between 0 and 1. So it might be 1%, it might be 10%, it might be 50%, but it's fixed. I'm not going to let it go to 0 as I pass to my scaling limit. And this just corresponds to the idea that your local population density is not going to be infinite. And then we ask, well, what's the chance that the ancestral selection graph is going to look different on these spatial and temporal scales? So I've set n for 1 over mu to save me having to say 1 over mu all the time. So if I'm looking at time scales 1 over mu times t, so n times t, and space scales square root of n times x, when will I be able to see that my ancestral selection graph is different from what I'd have seen under neutrality? In other words, when will I actually detect any of those branches? Remember, I'm looking, whenever I have a selective event, I get a branch in my ancestral selection graph. And it turns out to depend on dimensions. So let's have a look in high dimensions first. I know not many populations live in at least 3 dimensions, but it's the easiest case to deal with. So if your dimension is bigger or equal to 3, when we get to one of these branching events, we have 2 parents. And those parents are born at order 1 apart, and with positive probability, they're going to get far enough apart that they can no longer be encompassed by a single event. And then there's positive probability, because in dimensions bigger or equal to 3, random walk is transient. There's positive probability they will actually escape from one another and never see each other again. And in that case, I'm actually going to be able to detect that there is a difference. I do have a difference between my ancestral selection graph and my just ordinary coalescent. So in order to see a difference, I just need to see order 1 of those branching events. And that means that my selection coefficient has to be on order 1 over n. Because remember, we're looking at timescales n times t. And these come in at rate s. So I just need the selection and the mutation to be on the same order, exactly as if there were no spatial structure. Right? In 2 dimensions, it's slightly different, because random walk in 2 dimensions is recurrent. So if I start 2 random walks at a distance order 1 from each other, they will come back and see each other again. They'll come back and meet again, and they'll eventually coalesce. So the real question is, are they going to be able to get as far apart as the square root of n before they come back and coalesce? And for a single pair of random walkers, the chance of that is order 1 over log n. So I'm going to have many more branches. I'm going to have order of log n branches before I actually see any of these branching events when I'm looking out at scales of order root n. If they don't get to order root n, I'm just not going to see them, because that's the spatial scale over which I'm looking at things. So that means that my selection coefficient has to be quite a lot bigger if I'm going to see a distinction between the ancestral selection graph and just ordinary neutrality. So if I'm going to be able to detect selection, it's going to have to be stronger. And this is really the genetic drift. It's really because when in 2 dimensions, there's just not enough space. So even a selectively advantageous individual finds himself or herself competing with close family and behaving almost as if in neutrality much sooner. Okay? So this is 2 dimensions. 1 dimension, things are even worse, because the chance that 2 one-dimensional random walkers get to distance root n before they come back together is on the order of 1 over root n. So our ability to actually see anything is highly dependent on dimension, which I think is actually quite interesting. Okay, I've put the scaling limits 1. It's nice to make contact with with sort of more classical models. Fisher was always right at some level. It turns out that if we allow the density of our population to go to infinity, which corresponds to rescaling that impact parameter, that u, to go to 0, then we can obtain the standard classical Fisher-KPP equation. And in 1 dimension, you can even put a noise term in there. This alpha is just because one of the things you can do with this model is that you can say, well, how about having not just these small-scale things, which I'm thinking of as modeling sort of local reproduction events, but how about putting large-scale events in as well, large-scale extinction recolonization type events? And it turns out if you do that in the right way, then you can obtain a slightly different scaling limit in which the Laplacian is replaced by what's called a symmetric stable process. And this equation has been studied reasonably extensively, especially in the physics literature. Okay, I just wanted to put this, I'm just writing up the paper on this, is one of the reasons I put this slide in, but I just wanted to show we can get back the standard Fisher-KPP equation.
But can you do it with the noise term,
Yeah.
with the alpha?
Yeah, but it's always the same noise term. And that's what's interesting, and that's because and also this same term here. So you don't see the large-scale events. Remembering we're letting the population density go to infinity, the large-scale events don't feature in either the selective term or the genetic drift. And this is essentially because the large-scale events don't fall fast enough. They're not able to beat the small-scale events in turning up in here. So all they do is they move individuals around essentially one at a time, which is at first sight slightly surprising, and then you think a bit harder and it becomes clearer why it's true. You can do, if you try to do things with only small neighborhood size, you get a completely different result. Okay, so this is all mathematical toys, right? So I just wanted to show you some circumstantial evidence that the kind of genealogies that we would write down for this model may not be a million miles away from the kind of genealogies that you'd actually see if you were looking at a real population. So this was the easiest piece of data for me to get my hands on. Kevin Foster from Oxford sent me this picture. It's Pseudomonas aeruginosa, and the Pseudomonas are two different colours, green and blue, I believe. And what's happened is at time 0, a nutrient-infused dish has been seeded with a mixture of the two strains, which are equally fit, but they're expanding outwards. Now, one way you can think of a population which is expanding outwards is of individuals which are actually fit. They've got there's some selective advantage of being, how do I put this? You can think of range expansion as an extreme case of selective advantage. In nature, you might think that you have a population invading a less fit population. Here we've got the extreme case of an infinite selective advantage in the sense that you're moving into empty space. But you can use the same models to model it, okay? So what you see here is, you don't get the whole genealogy, of course you don't, but you do at least see some caricature of what's going on. So for example, all these guys out here must be descended from some blue guy back here, right? So you at least get some idea of what the genealogy is by looking at these stainings. Yep?
For the big blue chunks that are completely embedded in green chunks, how do those come to be? Is it just like a single blue lineage that didn't start expanding?
Like here, yeah, you just can't quite see it. It is there, but it's very rare.
Well,
and
do you think it's a threshold thing, that it just like crosses a threshold and then becomes a big chunk?
It's just stochasticity. I mean, you're sitting on the edge. So if you happen to be lucky enough to have made it to the edge of this kind of a wave, you've got a reasonable. You're just trying to spread out. If you manage to get ahead of the pack, you spread out really quite quickly. And it's just someone here has been really lucky. And it's just chance, because you've got an awful lot of trials around here. There are an awful lot of bacterium on this. So it's like doing. It's great in a way because you're doing probability theory when you've got an essentially infinitely big sample. And so you see everything that could happen will happen. Okay, so that's what bacteria do, and that's what simulating our model does, which doesn't look too bad. And in fact, we produced that before Kevin sent us this. And it's just fortuitous that he chose the right colours, bless him. So I cheated, but I didn't cheat in the way that you expected me to cheat. So of course I cheated to get a picture that looks quite as good as this, or either Jerome cheated. But he didn't cheat by doing lots and lots and lots of repetitions of the simulations till we got the right one, because as I say, we had this before we had that. But what he did do, and that's the actual outcome of the simulation, and because it's so sharp, it doesn't look so good. So what he did was, being a computer scientist, he knows these things. There is software which will make a sharp image look as though you didn't have quite such a high resolution camera. And so all he did was he replaced the sharp image by the blurred image, and it looked much better. But there's a serious point here, and the serious point is that we're really not trying to capture exactly what's going on in these bacterial colonies. We're trying to capture broad features, and I think this is circumstantial evidence, admittedly, but circumstantial evidence that we are capturing some broad features, because that is actually what the bacteria are doing. And we're not modelling that, we're not pretending to model that, but we are capturing somehow some of the broad features. And in spite of having started late, I'm only finishing a little bit late. Thank you.
Questions? Yes.
Is it necessary to have like a multiple merger thing going on to...
Yeah, you can't get... So if you want consistent forwards and backwards in time models, you have to have multiple measures of coalescence. That's right. Yeah. But actually, in practice, the sample from far enough apart, as you mentioned, you never see multiple merger anyway, so you don't need to worry about it. Because it involves string randomness.
How does it vary with the size of the model?
Ah, well, that's actually how we got the right number of sectors. So, I said we'd already got this. In this case, the number of sectors that we have is just a ratio of the initial droplet and the size of the ball. It's a deterministic function of those two, essentially. I mean, it's not deterministic; it's random, but it's very close to deterministic. Yeah, it's a scaling. I mean, but one of the reasons for allowing different sizes of ball was because we were initially interested in including these large-scale extinction recolonization events because we were interested in incorporating some of the demographic history into things. So in principle, one of the more serious things that you could in principle do with this model is that you could do simulations conditional upon quite complex demographic histories. So you could put in ice ages and things into your simulation. It doesn't have to be balls, it can be any shape you like. And I think Nick had this idea, he wanted to debunk, am I allowed to say this? He wanted to debunk phylogeography by actually doing things conditional on, simulating conditional on the history.
So if I'm thinking about this as hitchhiking, I'm thinking about these different sectors corresponding to different recombinant haplotypes which have surfed along with the allele, right? So in that case, do I think about the time scale over which I'm looking at, instead of being parameterized by one on the mutation rates, being one on the recombination rates?
Well, in this case, they're not. I mean, there's no recombination going on in this picture, right?
No, a bit more
But
if
if
I'm
you
trying
were
to think
doing
about
that-
the...
Yeah, that's right. So if you have to have both, right? So the mutation rate is telling you whether or not you've got a chance of seeing anything, and then the recombination rate will tell you how far away from the selected locus you can go and still expect to see something. You still need the mutation rate in there somewhere, because if there's no mutations on your genealogy, you're not going to see it.
But the mutations can be deeper in time, right? So they can be
They could be
alleles
pre-
which have been shuffled around, so...
Sure. But if it's too far back, it's not going to give you a signal.
There should be some more.
Okay, so thanks to Alison and to everyone this afternoon.
Thank you.